Dear learners, welcome to the NPTEL online certification course on novel drug delivery systems. I am Dr. Satish Devanapelli, working as an assistant professor at Institute of Chemical Technology in the Department of Pharmaceutical Sciences and Technology, Mumbai. So, in our novel drug delivery systems, today we are going to discuss on the topic Introduction to Novel Drug Delivery Systems. So, here I am going to discuss on various topics. So, here today we are going to learn on fundamentals of drug delivery systems. So, these are my flow of presentation, and today I am going to discuss on what are the drug delivery systems, and after that we are going to see the difference between conventional versus drug delivery systems, and further we are going to see the evaluation of the drug delivery systems, and further we will see what are the terminologies used in drug delivery systems. Also we see few definitions. Then we will also look what are the advantages and disadvantages of drug delivery systems, and then finally we will end up our presentation with generations of drug delivery systems. So, as we know, so developing drugs for a particular disease, it is one of the major challenging, because we know the process of drug discovery development. So, this can be split into various phases, so which are nothing but drug discovery and the development. In the drug discovery process, before the development and the clinical trials, so we need to identify the right target or right pathological system to stop the diseases. So, in this process, so various scientists will involve for the know the pathological systems of any diseases, and we will not end up at that stage. Further, so many scientists will involve in designing of the various molecules. So, various design approaches, scientists will discover the molecules, and after that will, after performing into various in vitro studies and then preclinical studies, the targeted molecules will reach into the clinical phases after performing the safety and the toxicity studies on the animals. And then in phase 1, phase 2, phase 3, we will understand what is the nature of the drug on human beings in terms of safety, efficacy, and other parameters need to be considered. After performance of phase trials, clinical trials, so molecules will undergo for the drug approval from the regulatory body. So, once through the NDA, new drug approval process, after completion of that, so molecules will after 2 to 3 years of the reviewing of from the regulatory bodies, then they will give the approval for the marketing. And the story will not end that stage. So, here we are today going to discuss on the drug delivery systems. So far we have discussed the drug discovery development. So, making or developing or inventing of new molecules is one of the major challenging. It takes lot of time for the development and for the approval. And when you develop any formulation from that API or new chemical entity, so that will undergo lot of problems in terms of formulation challenges. As we know, as per the BCS classification, biopharmaceutical classification system, so drugs can be classified into 4 categories. So we know that class 2 and class 4 drugs suffers with the low solubility. So these drugs need to be converted into the solubilized format or otherwise which need to be converted into the right dosage form, so that can enhance the solubility and the permeability that will leads to improve the bioavailability of the drugs in the human beings. So we can understand from this what are the challenges happening in the drug discovery development. So designing of drug products or drug molecules is one of the major challenging for a particular disease, and the drug should be reached into the target site also is one of the major problem in the treatment. So here, yes, we know so many conventional formulations as I mentioned here, like converting of new chemical entities API into the dosage form is one of the major challenge. So from that we will make a tablets, capsules, syrups and ointments and injections, which are the conventional formulations. But when you are going for the diseases like brain diseases, like cancer diseases, some other type of the infectious diseases, so the conventional systems are not effective. So to avoid the limitations in terms of the efficacy, in terms of the poor biodistribution and the side effects and the lack of selectivity, we need to look for the high standard systems, which are nothing but drug delivery systems. So with the drug delivery systems, we can convert a conventional formulations into the novel drug delivery systems. So delivering therapeutic compound to the target site is the one of the major problem in the treatment of many diseases. So making a formulation as a conventional formulation will not be effective for the treatment. So such cases we need to convert our active molecules into the novel drug delivery systems by using various approaches. All these drawbacks can be overcome, and that can be improved by using the only with the drug delivery systems. Now with this picture you will understand, if you give for a cancer patient with any type of the conventional formulation, so the drug will distribute throughout the body into various organs. So this leads to the toxicity in the patients, and which is non-selective, and the distribution is not proper, and definitely it will not show the efficacy. So to overcome this, a formulation or a drug that must be translated or converted into drug delivery system, so that we can reduce the side effects and other limitations. Now you can see here, after injection, this is the tumor target site. You will not see any type of drug presence or accumulation in the tumor site. So to overcome this, we must convert our active pharmaceutical ingredients into the drug delivery systems. So here, what is the drug delivery system? So the definition suggests that these are the technologies. We use lot of technologies to convert into the active ingredient from conventional formulation to the drug delivery systems. So these are the technologies and the formulation approaches and the methods used to transport pharmaceutical compounds in the body to achieve therapeutic effect in a safe and controlled manner. So here we work on the various types of the the methodologies and the processes and the materials and that need to be treated with our drugs, then that will convert into the as an effective drug delivery systems. So, that will show the safe and in a controlled manner. So, the main aim of NDDS or novel drug delivery system is to improve the efficacy, safety, bioavailability and the patient compliance of the drugs by offering controlled, targeted, sustained release of the drugs after the administration into the human beings. Now, here I have mentioned highlighted some of the drug delivery systems. So, we need to convert our conventional formulations into the drug delivery systems. The active ingredient will be same, the dose will be reduced, the efficacy will be improved and the safety also will be improved. So, here by using the modified formulation, we can control the release by using various types of the polymers and the carriers and other technologies. We can also use even we can also convert the as well as capsules also by using the various types of the polymeric systems. And we can also convert the drugs into the microneedles to show the target specific delivery. We can also convert your drugs into the nano formulations by using various types of the nanotechnological approaches. With that, we can enhance the solubility as well as permeability, will show the high efficacy in the body. And yes, we can also use injections that we can convert into the long acting injectables by using various types of the polymers and other ingredients. We can also make the site specific type of the delivery systems for the transdermal. We can use even nano carriers into the patches that will also show the efficacy and the safety in the human beings. So, as well as we can use pulsatile delivery systems. So, these are here we are limited ourselves to see the few of the drug delivery systems. In further our presentation, we are going to discuss on various drug delivery systems. Now, here you can see these are the non-specific targeted delivery and this case is the targeted delivery systems. So, here you can see the drug accumulation in the tumor site. So, with this we can improve the patient compliance and improve the safety and efficacy in the human beings. Now, here we will understand what are the differences between conventional drug delivery systems and the novel drug delivery systems. So, here in this we are going to see the various aspects going to compare with the conventional systems and the novel drug delivery systems. Now, we know these are the traditional methods and these are some advanced engineering systems to improve the delivery of the formulations. So, here we use various strategies and the materials and we need to treat our drug with the various technologies. Now, release profile, definitely we know that in terms of conventional tablets and the capsules and other systems, so yeah, definitely your drug will release immediately, that will not show the long term effect. Whereas, in novel drug delivery you can go with controlled and sustained and targeted delivery with modifications. And targetability, very difficult, it is not possible for the in case of conventional. Yes, by using various types of biological molecules and you can use ligands and also you can use polymers. With that we can specifically reach into the target site, especially in case of tumors we can use pH sensitive, temperature sensitive polymers, they will reach into the target site. And you can also use various ligands, for example folate as a ligand we can use it to target the folate overexpressed cancer cells, for example breast cancer. So, yes, in terms of formulation these are the examples of formulations and here we can use various types of the nanoparticles, liposomes and microneedles and implants. With that we can improve the safety efficacy of the formulations. Dosing frequency, yes, we need to give each interval, but when you convert into the NDDS formulation, we can reduce the frequency of the administration, that will show the long acting. And patient complaint is very, very less, because you need to give multiple doses, whereas here the patient complaint is very, very high. If you convert your formulations from injection to the as a nano formulation or some other targeting systems, definitely your patient complaints will increase. And also we can reduce the side effects by converting or by reducing the dose of the drugs. And yes, this is again we already discussed, the higher due to systemic exposure, because you need to give multiple doses here, right, your dose size will increase, whereas here yes, we can expect even 7 days of the activity. And also we can also see some of the market products which suggest that it will release the drug till 1 month also. So that is the beauty of the drug delivery systems. So we can also reduce the localized action. And stability, yes, it is not possible when you want to use any biological molecules or any temperature and pH sensitive molecules, you cannot go ahead with the conventional systems, because which are not stable, the drug will degrade in the body. But whereas here you can improve the stability at the outside environment as well as in the body. So these polymers which will protect from the all type of enzymatical, pH and other types of the degradations in the body. With that we can improve the stability as well as bioavailability. And limitations, yes, poor targeting and the fluctuation of the drug in the release, and but here the cost and the complexity and regulatory hurdles are there, because here the reproducibility is one of the major issue, and also the unestablished the regulatory guidelines are there for the novel drug delivery systems. But whereas in case of tablets or other formulations, we have known and established regulatory guidelines. So that is also one of the limitation in case of the novel drug delivery systems. These are some examples of the paracetamol tablets and injection, whereas yes, liposomal doxorubicin and the transdermal patches and mRNA lipid nanoparticles, which are the example for the novel drug delivery systems. So here we can see, as I mentioned in last my presentation, with this we can expect 12 hours of the release. This is a one brand name Spansul. So with that you can, it is a capsule technology. With this you can expect the 12 hours of release from the drug delivery system. Lupron depot and long acting injectables and implants, you can also expect in the release for months. So this is the evaluation of the drug delivery systems. Here I have shown very limited example, but you can go through this reference. You will get all type of information in this link. And so these are the various types of delivery systems. Here you can see the pegylated proteins and the doxorubicin in pegylated liposomes, pegylated naloxone, and even the biological molecule like siRNA. And nowadays we know all during the COVID also. So we know that the vaccine which comes in the mRNA vaccine. So we know the next generation is entirely will depend on the small biological molecules. So see for this case, so safety and the improvement in terms of the patient complaints is very, very important. So we need to consider while developing the developing of any type of novel drug delivery system, we need to consider all the physicochemical properties of your drugs as well as biological molecules. Based on, you need to select right material, and we need to process through right technology. So here we are going to discuss the common terminologies used in the drug delivery systems. So far we have discussed few terms, but here also we will see some terms which may come during my presentations or when you want to learn as in a drug delivery systems. So these words are used repeatedly. So the first word is the control release, and the prolonged release, sustained or slow release, long acting release, delayed action, gradual release, modified release, and then prolonged action and the pulsatile release. So, these are synonymously used in the drug delivery systems. So, here we will see there are the two definitions, that is controlled drug delivery and the sustained drug delivery. So, here sustained drug delivery allows delivery of the drug at a predetermined rate. So, this is very important. So, the drug will release at a predetermined rate for a locally and systematically for a specific period of time. So, here the drug will release the at particular rate and for particular time. So, it will not depend on the concentration of the drug present in the drug delivery system. So, this is nothing but zero order release. So, the drug will release over the time irrespective of the concentration. Such release is called as zero order release. So, zero order controlled drug release system also can be developed by converting of the conventional formulation into the controlled drug delivery systems. So, here another definition, sustained drug delivery, which allows delivery of specific drug at programmed rate that leads to drug delivery for a prolonged period of time. So, here the drug will release continuously, nothing but it is shows the prolonged release, and here the concentration, it is depends on the concentration. So, here how it works, we can see here the drug released immediately, that is nothing but initial dose, which is important for the therapeutic response, and more promptly the remaining maintenance dose is then released slowly, thereby achieving therapeutic level which is prolonged. So, initially the certain amount of drug will release, that is a initial dose, then further it will maintain for longer time. So, which is called as the sustained drug delivery, or you can say delayed or prolonged drug release. So, these are the some of the common terminology which are used in the drug delivery systems. So, now with this figure you will understand what are the importance of the drug delivery systems. Now, you can see here, so when you are first we need to try to understand what are this graph. Okay. So, this will be plotted between the time versus plasma drug concentration. So, here this is a x axis and this is a y axis. So, this is a theoretical approach to understand what is the importance of drug delivery systems. So, here when first we will see the single dose of immediate release formulation, or you can say the conventional formulation, this one. So, we need to give the dose, right, and how it works, you can see with these lines. So, here again we need to understand few factors here, that is minimum safe concentration, minimum effective concentration. If your drug want to be effective, your drug should be reach the above this concentration. So, that is called as therapeutic range. The minimum concentration required to show the efficacy or therapeutic effect on that particular disease. So, that is called as MEC, minimum effective, and the another one is minimum safe concentration. So, below this level your drug will be very safe till this concentration in the blood or plasma. So, above this it will show the toxicity. So, after this, for example, we know that paracetamol will induce the liver toxicity. So, that means when you take high dose for longer days, your liver will damage, that is called as toxicity. So, here when you take conventional formulations, at each time interval you need to take a dose, multiple doses to maintain therapeutic range. So, to maintain this range, we need to take various doses. So, to avoid this, this leads to the patient incompliance, and we need to give frequent doses, and also it will leads to the toxicity. So, to avoid this, our formulation need to be converted into the various types of the drug delivery systems. So, today we are going to see two things. So, first is, as we discussed, zero order control release formulations. So, now you can see, so the drug is released and then it showing, as we discussed in the definition in previous slide, in that will show the predetermined rate for a specific period of time. So, that is called as zero order release. Whereas sustained release, so it depends on the concentration, and that is first order release. Now, initially the dose will release and then it will sustain for longer time. So, that is called as sustained release. Now, your dose frequency will be reduced here and patient compliance will also increase, and you can see the therapeutic level it will maintain for longer time. So, this is the difference between the sustained and the conventional release formulation. So, these are the terminologies we will use in learning of drug delivery systems. So, with this today we are going to stop our lecture. So far we have discussed what are the drug delivery systems, then we have discussed what are the conventional and novel drug delivery systems, we have seen the differences, and then further we have seen the terminologies used in the drug delivery systems. So, today we are going to stop here, and in further lectures we are going to discuss on the various topics and the fundamentals of the drug delivery systems. Thank you so much.